Thanks, Dan. And thank you all for having me. I got to do this presentation with the chapter in Silver City and the one in Albuquerque. So I'm excited that more plant people are sort of interested in this topic. It's kind of unfortunate. It's like only bug people that are do the primary gall research, but so much is plant. You know, this is not just the insect. The plant has it's a 50-50 relationship. So I'm excited to get more people interested. Um, so just a quick, Jan gave my background just fine, but I also was just employed at for the Center for Species Survival in New Mexico, which is based out of the New Mexico Biopark in Albuquerque. So now I work in insect conservation. So mostly regional insect conservation and work on endangered pollinators. So, you know, that I'm talking about galls today, but if anyone wants to talk to me about invertebrate pollinator conservation, that's sort of also my focus. Okay, so today I'm going to split the talk into 3 sections with the goal of explaining what galls are and why I think they're cool. Show you some common ones that you might want to start recognizing out in New Mexico in general, and then I'm going to share a bit about my research that I did for my thesis just as an example of the kinds of questions we can ask about galls, and then hopefully there'll be enough time for some questions at the end. Okay, first of all, what am I even talking about? So a lot of people compare them to tumors because they are these abnormal growths that occur on plants that are a proliferation of cells. So it seems like a good comparison, but the distinction is that they're specifically caused by an outside organism. So we call that the gall inducer. And they can do this because they use, well, the reason that the inducer induces the gall is to create a home for their offspring to live inside that is protected by plant structure and also has nutritious cells on the inside to feed the developing offspring. So it's a multifunctional structure. And one of the weirdest things about galls is that they're really mysterious, which is why I got into it and why a lot of people got into it, is that we don't have a good enough understanding of how this interaction works. Nobody has been able to replicate this in the lab. We can't synthesize that really specific interaction that occurs to make these structures. So I think that's really cool. Every time there's something that remains a mystery in science, I think that's awesome, and hopefully you do too. So the habit of gall formation is also interesting because a lot of different organisms have evolved to adopt this habit. So there's bacteria, viruses, and fungi that all create galls on plants. And there's also invertebrates that do it, like nematodes and mites. But most of what I do is insect galls. And so I'm going to mostly tell you about insect galls today. Fungus and bacterial galls are fairly common where we are because they are more associated with conifers. So on conifers, a lot of times you'll see fungal galls like that one. But you're probably like, well, that looks like lots of things that I see on trees. And so just as a quick, I'm sure you all understand these words better than I do, but just to make a distinction, I'm not talking about burls, cankers today. So the distinction is, from my understanding, is that burls are deformities that are sort of like a callus or sort of a proliferation of deformed tissue, but they're created by the plant's own immune system. And so that could be a response for an infection or something like that. But a gall has to have an invading organism that induces the growth of that nutrient-dense tissue. And so it's difficult to tell apart in nature. And unfortunately, I can't always tell you which is which, but insect galls are really easy to recognize. Fungal galls are a little bit more challenging. So, right, okay, like I said, also nematodes and mites, so there's animals that make galls. And insect galls are the most species rich and are super, super diverse and have a ton of morphological characters that we don't see in other gall types. So things like really bright colors that you don't see anywhere else on the plant, things like hairs, spines like this on a non-spiny plant. This looks like a pine cone sort of structure, or a rosette of some sort. This is on a willow. And so they can create these really unique structures, and they're super diverse. They come in all shapes and sizes. So how does it work? An insect like this one, this is a female. She's inserting her egg, that's her ovipositor there, into her plant, into her host plant. And at that site where her egg was inserted, a growth forms. And so this is the gall of that fly. Anyway, this is what it looks like on the inside. So if I cut one of these in half, this is the part of the stem. This is the stem, and this is where the egg was inserted. The egg hatched, the larvae fed within this gallery while the growth formed. And then once it was formed, it crawled inside, and there's the larva now. And you can see that it's eating this tissue on the inside, these are really protein-rich cells. So these cells that line the inside of the gall are actually specialized so that they're highly nutritious for the inducer to eat, versus the cells on the outside, which are more structural. And so eventually this larva will eat so much that it fills almost the entire inside of this gall, and then it will pupate and emerge as an adult so it can mate out in the world, and then the process starts all over again. And I want to make it clear that this is a super unique habit. This isn't mirrored other places in nature. There's no other, these gall inducers are unique in that there's no other animal habit that we know of that makes a plant build something for it. It's really, you can't go out to your apple tree and make your apple tree grow an apple for you. It's really cool. So that's like the mystery. How does it get the plant to make this structure that the plant has no ability to make on its own? The only gall which we do have some sort of an idea of how it works are bacterial galls. And we understand that process because bacterial galls are really simple. They are a lot like tumors in that they're like homogeneous all the way through. So this is a bacterial gall sort of cut in half. And the reason that their process for making a gall is that it implants a piece of DNA, that bacteria implants a piece of DNA into the genome of the plant. And so then when the plant is replicating its cells, that piece of DNA from the bacteria also gets replicated, and it results in the growth of these structures. And so that's really cool. That plasmid that is in that genome encodes for the synthesis of hormones. So hormones like cytokinin and auxin that drive the plant to create cells, to create new tissue. But then also it creates something called opines, which is a carbon source that only the bacteria can metabolize and the plant can't. So this encourages the growth of that bacteria specifically. So that's all we know. That's the extent of our knowledge of how galling works. So the difference between that gall, the bacterial gall, and an insect gall is that insect galls have, like I was telling you earlier, they have specialized nutritious tissue on the inside, and then some sort of external tissue on the outside, which is always very different in structure and results in the high diversity that we see. And so this is not simple. This is like many different cell types that are being created by this invading organism. So one theory about how galling works, which we have pretty good evidence for, is venom. So these are wasps. A lot of galls are created by wasps. So there's a couple different wasp families that are gallers. All wasps have venom, but they're not always a venom for animals. Sometimes it could be a venom for plants. And so this is an image of a venom gland, this really long structure here. And so in these teeny tiny little wasps, we know that venom is super important to how the gall is created. But what we don't know is how to pull apart that venom cocktail into its constituent parts. So we don't actually know every single chemical component to that venom. So great, we know the venom is playing a role, but we can't replicate it. We don't know how to make it ourselves. Another probable method of inducing galls is salivary secretion. So a lot of galling insects don't have venom, like flies and midges. Those, they don't produce venom. But what we find is that, like the image I showed you before, the gall actually appears after the egg hatches and the larva starts eating. So it might be the larva is eating and secreting something from its salivary glands that then the plant is responding to. And this image has, you can see this bright green is plant hormone that is seems to be really high in the salivary glands of some of these galling insects. So that seems like a really excellent area of research as well. As you might guess, a venom gland on a wasp is super small. Think about how small a salivary gland of a larval insect might be. So it's really challenging to research. So we only have one gene that we've been able to identify is responsible for galling, and it's the only gene we have at all is just a gene that determines if the gall is going to be green or red in one species, and this is an aphid. And so that kind of is just an example of how much information we have to learn about this process. There's probably hundreds, maybe thousands of genes that are responsible for what the gall looks like, and we've only been able to find one so far. Another theory for a long time was that because we understand how bacterial galls are formed, maybe the insect has a bacteria in its body that helps him form a gall. But so far, there hasn't been any good evidence to support that. This is just a map of different insect orders and their bacterial communities in this sort of 2D space. And you can see that ones that create galls in the triangles and ones that don't create galls in the squares, there's no pattern there. They're all over the place. So it doesn't seem to be an association with microbes. But who knows? Maybe we just haven't found the right system yet. Okay, so you're probably wondering, well, which insects make galls? You're just talking about insects in general. I think this is pretty fascinating, but it's evolved independently in 6 different insect orders. Independently. This wasn't just one mutation that happened and then a bunch of other insects. This habit has shown up many different times, which probably indicates to us that it's a good habit to have. It's advantageous. It creates this protective structure. It creates food. And so the most common galls are wasps and sawflies, which are in Hymenoptera, midges and flies, which are in Diptera. And then there's also a few hemipterans, which are like aphids, things like that. There's a couple moths, there's a few beetles, and then there's thrips, which are kind of they're such a small insect that people rarely see them, but they're all over the place. And then, like I said before, mites also create galls, but mites are an arachnid and not an insect. So, but another example of how this keeps evolving, and I think that's really fascinating. And then another common question I always get asked is, well, which plants have galls? And what's kind of interesting is that every time somebody asks me, hey, does this have a gall? Does this have a gall? Like just any random plant that they throw at me, oftentimes I'm like, I'm not sure. And I go check, and the answer is almost always yes, which is fascinating. So I found listings for galls on some like unexpected plant groups like aloes, orchids, bromeliads, sedum, cycads, equisedum. But they're definitely grouped. They're most abundant in just 4 different plant families, which are plant families that are super successful. They're widespread, but you'll most often find galls on Fagaceae and Asteraceae, and then there's a few that are specialized to willows, and a few that are on various Rosaceae plants as well. Okay, this is a question that I think I get asked every single time I talk about galls. Is like, well, is it a pest? Like, because it definitely looks like something is happening, right? That might be harming the gall, especially when we compare it to a tumor. That seems like it could be bad for the gall. So the short answer is yes, because it's a parasitic relationship. The insect is taking advantage of the plant and getting it to create new tissue for it. And so there is energy that's being used to support this parasite. But there's sort of a spectrum on how much it might be affecting the plant. So this is sort of an example of when it can hurt the plant and be pretty impactful. So this is a blueberry stem gall wasp. If anybody has spent time in Michigan or that area in their blueberry agriculture there, this is a huge pest problem. And this is one of very few galling pests that we have. But the reason it's bad for the plant is that this gall appears on the stem like this, and it starts to curl the stem. And eventually, because the larva is done feeding, it no longer requires that energy and water is moved to the gall. And so the stem at the end of the gall comes off. And so the tip of that branch dies. And then, as you can imagine, that results in there not being as many flowers and not as many fruit. And the other problem is that if you've ever seen blueberry harvesters, they're kind of like cones that come through to pull the fruit out. They just get chock full of these galls instead because they're bigger than blueberries and they're super hard. This is a wasp. However, the vast majority of galls, it's unclear if the plant is being hurt or not in terms of its reproduction, in terms of its longevity. So I like to bring this picture all the time. This is the most heavily galled rabbit brush I ever came across in my research. And this is during winter, so there are no leaves on it anyway, but which allows me to see how many galls there are on it because all the leaves are gone. And there's literally thousands. Like this is a close-up. There's 20 per stem. It's amazing. This is in El Malpais National Monument. So it seems that it's similar to a lot of human parasites. If the host is healthy and has access to water and nutrients, then the parasite living on it is going to have minimal impact. But, that remains to be seen. It's really difficult to tell whether or not this is, because in this case, this is also a stem gall, but it comes from the side. It grows off the side of the stem from a node where a leaf would grow. And so the the end of the stems are not affected at all. And unlike the blueberry, which kills the end of the stem, these are not killed, and this plant will be completely in flower at the end of the season. So, and then I always like to bring this up, that there's actually some gall mutualism. So there's some cases where there's a benefit. So you might know about fig wasps. Those are technically gall wasps because they're modifying the plant tissue inside of the developing fig. And so that's a perfect example of a mutualism. That fig wasp is actually pollinating the plant while it's creating its gall. There's another cool study I like to talk about. So the fig is not a gall, but on the inside, there is a mixture of fruits and galls. So some of them are completely galled, and some of them are mature fruits. It's kind of fascinating. And I could tell you more about fig wasps if you want another time. They're kind of, it's a great example of coevolution, right? The ficus genus and these wasps just kind of exploded together in terms of diversity. But another example that I like to bring up is there was this study where they found that there was a bunch of invasive goats in this place in Spain, and I think it was tarragon. I should write down which plant it is. There's galls on them, and the goats were eating the plants that didn't have galls on them and leaving the galled ones alone. And so they looked into it and found that tannin concentration in the gall was like 4 times the amount it was in the leaves. So they were distasteful to herbivores that would come around. And these galls are bright red as well. So there's sort of, can't be proven, but it's sort of a theory. Hey, maybe the plant actually benefits from having the galler on there because the galler is not killing the plant, but it is keeping away the herbivore mammals that could kill the plant. So there's definitely some interesting questions there. Humans have used galls for centuries. Native people here in North America, but in Europe and China, they loved galls and using them. There's some that you can eat. So that I read about a famine where people ate these galls, added those to their diet so that they could, get through the famine. And the Declaration of Independence is written in gall ink, as well as a lot of other documents like the Magna Carta and things like that. So gall ink was really popular and valuable for a long time because, like I was talking about, the tannins get concentrated in a lot of these galls. And so you can harvest the galls off of oak trees, and then you get this super tannin-rich liquid, and you can use that as a dye and a ink. And I've tried it before, and I didn't do a great job, but I was able to write with it. It was really chunky. I'm sure there's like a better way to do it. Okay. I'm going to now tell you about some galls that you might see in this. I kind of just did it for the state. This isn't like specific to the Sangre de Cristos or anything like that. So, but I gathered some photos just so people might start recognizing some of these really common ones. So on the scrub oaks that we have, there's a lot of oak apples, which are these pretty large. So they can be as big as this. In New Mexico, we have pretty small ones that are about that big. And they do kind of look like little apples. And it's confusing because you're like, wait, I thought that was an oak. And so oak apples are caused by wasps in the genus Etrusca. And what's the cool thing about oak apples is their interior design. So this sort of outer shell is pretty flimsy. And, you see these galls and they're about that big. The wasp itself is itty bitty bitty. So why does it need all of that space? Well, it actually wants all of this empty space around it. And the wasp only lives in this tiny little ball on the inside. And there's a few different theories for why it does this that I can go into detail later. But basically, it's trying to insulate itself from the environment and from predators. There's things that actually want to eat that developing juicy larva in there. So it makes a structure that's way bigger than it needs and then lives just in the center of it. Yeah, oak apples have just one. Yeah, it's pretty crazy. and oftentimes you see them in pretty high abundance when they're on the oaks. So Gamble oak also has a lot of galls on it. So that's the same one, the Etrusca there. So Etrusca is on both Gamble oak and the scrub oak. Yeah. I like this picture because there's actually 3 different galls in one picture. So there's a stem galler here, and there's a little cup gall right there next to a bunch of these, we call them pea galls, on the bottom of that leaf. So that's a really cool picture. So 3 different species from 3 different genera. Those are all wasps. So as you might be beginning to suspect, wasps like oaks. So if you find an oak gall, it's almost for sure a wasp. There's ones that are really hard, and then there's ones that are really soft and fluffy, and you can crush them really easy. Yes, this is actually a mite, this one here. So that's one oak gall that isn't a wasp. Those are super common. They just look like little fuzzy tips. This is arroyo willow, right? Coyote willow. And coyote willow has a lot of midge galls on it. So this one is super common, that creates these little rosettes. And then this one is also really common. And this is kind of another example of how the stem dies beyond the gall. And then here's one here where the stem doesn't die. The stem is just slightly expanded. And I like this picture because you might start to go, oh, how many times did I just not even know there was a gall there? This looks pretty similar to the normal stem, but you can see the emergence hole right there. Yeah, so this leaf dries and it just falls off, yes. And then the larva, the timing is such that once it falls off, the larvae they're done. They're not going to keep developing. And so that's when they pupate and turn into adults. And then the adults need to leave the gall so that they can mate. So they're not equivalent to the cocoon. The cocoon comes after. The cocoon is what you pupate inside. And so you can there's some gallers that pupate inside the gall. Most of them do because it's more protective to do that. But there's some that actually leave the gall and then pupate in the soil below the plant. So an answer to a lot of these questions is going to be sometimes yes, and sometimes it's this other thing, which is crazy because there's so many different insects we're talking about here.
Yeah, are they predator wasps or beneficial?
Well, so this is wasps that make galls. This is what they do. They're not predators. They don't predate on anything. They might pollinate by accident, but it's unlikely that that's major at all. So I am not going to say if they're good or bad wasps. These are wasps that this is their life. They make a gall, they develop in the plant, they come out, they mate. I don't know, that doesn't really answer your question. But they're not like well-known beneficial parasites. No. Yeah, because they don't eat other pest organisms. But I'll get to that. Will lead me to something in a second. Just some other willow galls. So a lot of willow galls, this is another midge, but a lot of willow galls are sawflies, which is a group of bugs that nobody ever talks about or hears about, and they're they're like ancient wasps. They're wasps that they evolved before wasps became predatory. So they're plant wasps, which is really cool. In German, they're just called Plantwaspen or something like that. But we call them sawflies because their ovipositor saws into the plant. So I really love sawfly willows, and one cool thing about them is that, willow sawflies, is that nobody does any research on them, and so there's a ton that are undescribed. So I just wrote unknown for this one because nobody knows what this species is. Cottonwoods have a lot of galls. Mostly they're all hemipterans, so like aphids. So aphids almost always have communal galls like this. So there's several that live in there. There's another mite that is really cool when you see it because it's bright pink on a cottonwood. Our pinions, our conifers also get galls, and they're often less flashy for some reason, but you can still find them. So this is two different kinds of midges that are just at the base of the needles, which is really cool. This is another unknown species. Somebody could describe it if they wanted to. We have a ton of these ephedra galls all over, at least down where I am, like in central and southern New Mexico. I don't know how much ephedra there is up here, but we've got a ton of these. They're all over the place. Those are midges as well. Creosote has a huge number and diversity of galls, probably because it's so old. And these midges, this one genus of midge, so Asphodyllia, all of these midges are in this genus, appears to have co-evolved with creosote. And so they make a ton of different galls just on creosote. And this one is super cool when you find it. I think they call this one the Medusa creosote gall, if I remember correctly. And then another, this might be too complicated, but probably some of you know how about there's these populations of creosote that are polyploid. So they have multiple copies of their chromosomes. So like in southern Nevada, there's a tetraploid creosote population. They found that you can actually tell which ones are diploid or polyploid based on which galls they have on them, meaning that this midge is adapting to that specific ploidy, like is responding to, has evolved to choose that plant instead of the other one, which I think is really fascinating. So all of these ones on the right are polyploid specific creosote galls. Saltbush has a lot of galls on it. These are also Asphodyllia midges. Sagebrush has a lot of midges on it. These are all in the genus Rhopalomyia. There's a lot of undescribed species in that genus as well. And then I'm going to end on rabbitbrush because that's what I did my research on, and it'll transition into that. So these are a few more Rhopalomyia midges that are on rabbitbrush. There's also some unknown ones that we have in pretty high abundance here in New Mexico. But I worked on these galls that are induced by a fly in the genus Asurina. And the two most common ones we find are this cotton ball looking one and then this smooth kind of resinous sticky one. And I don't remember if I have a map in here, but where we are right now is actually where the two of these meet. So down this sticky one is really abundant and common along the Rio Grande, sort of like down south. And then this cotton one is more of a mountain associated higher elevation species. And so the Rio Grande Gorge State Park is where these just occur right next to each other, totally happily. And these are the flies that create those galls. So that's going to lead me into talking a bit about the research I did, just as an example of like, what are some of the questions we can ask about these systems. Hopefully it doesn't get too dense. But so a cool thing about galls is that each gall inducer, so this fly is our gall inducer, right? She laid her egg here, and then this gall formed, and now the larvae is living inside. Each gall inducer is what's known as an ecosystem engineer, meaning that it creates a structure that actually serves as habitat for a bunch of other organisms. So galls host especially species-rich communities of all kinds of different arthropods. Even though they're really, really teeny tiny, there's actually a ton of different organisms that interact with that structure and rely on it to complete their life cycle. So
the larvae is developing inside it.
Yeah.
It's all going.
Yeah. So one example is parasitoid wasps. So you were asking about beneficial predator wasps. These are those. So there's a ton of parasitoids that eat galling wasps or any sort of gallers. So this is my fly larva here that has been parasitized. So it's sort of desiccated and basically like a shriveled up sleeping bag. And that's the wasp larva eating it, sucking the juices out of it. And then this is the same wasp as an adult. So pretty hardcore. They just the cool, we see a lot of these really complex structures of galls, and one theory as to why they are so complex on the outside and why we see so many characters like hairs and spines is that they're trying to protect against those parasitoid wasps that want to attack them. But then we have sort of the counter argument is that these wasps get really good at growing a really long ovipositor. So a stinger is the same thing as what lays the eggs. So this is their egg laying apparatus. So she comes across this gall, and her ovipositor is long enough that she can stick it all the way through and insert her egg into the center where the gall inducer lives. So that's this wasp there feeding on another desiccated corpse of my poor fly. So that's a pretty cool adaptation back and forth that appears to be happening. And so from these two gall species, these are all the different parasitoids that I was able to rear out of them just in northern New Mexico. And so that's, I think, a pretty good example of how diverse and huge these communities can be that these galls are supporting. And so what we don't know is how specific all of these parasitoids are to these galls. Can they only feed on those? Can they feed on other animals? We're not sure, but you can see there's a huge range. We have ones that have short ovipositors and ones that have long ovipositors. So there's some really interesting adaptations there. So what I wanted to do is I wanted to compare to see if there was a difference between the parasitoids that attacked this gall versus this gall, because if what we think about the external structure is true, that it might be protective as a defense, then you would assume that this one would have less parasitoids than this one, or at least different parasitoids, because you could also argue that the sort of sticky resinous substance that it creates on the outside, that could also be defensive, right? So I tested to see if they were lower, if they were different, and they were. So the communities are plotted here, and we got different communities, significantly different communities of parasitoids, and found that the one with the hairs on the outside did in fact have lower rate of parasitism, so supporting that might be a defensive adaptation. Another associate that I want to tell you about are what's called inquilins. So you might have heard that term in birds before, something like that, but that just describes an organism that is, so a parasitoid comes in, kills the host, right? But an inquilin comes in and starts eating the gall next to the host. So it's kind of stealing resources from the host, but it's not killing the host itself. So this is it would be where the inducer would live here, and then there's a bunch of inquilins on the outside. I like this picture because they basically turn the gall into like an apartment building, and then they all just sort of share it. And a lot of times the inducer can develop just fine, and so can the inquilin. So in this case, this gall, here you can see there's 2 emergence holes, meaning that both the fly and the inquilin weevil, in this case, emerged as adults and continued their life cycle. So they were able to share it peacefully. Yeah, and I really like how cute this little weevil is. They're very adorable to me. And then another cool inquilin that we found, we found a population of these cotton galls with these little pimple-like structures on the outside, which was sort of confusing to us. And when we cut them open, we found that there was an insect living in them. But this is where the inducer lives. That's the inducer pupa there. You can see how big it is. This is a completely different insect. And so upon closer inspection, each of these little bumps has a midge larva living inside of it. And these are galls. So this midge is galling a gall. It made a gall on top of a gall. So that was pretty exciting to find. And also they're really bright orange, which was super fun for us.
So they're creating their own gall in the original.
Yeah.
Wow.
And it might be that they require a gall in order, it might be that they can't make their gall anywhere else, that they need the other one in order to do it, because that tissue is different tissue than would be on the plant. Yeah, it's cool. And this is super rare. We've never found this before, so it was super exciting. And another thing is that you remember all those parasitoid wasps that were associated with the flies. There's a whole other group of parasitoids that were about 1/10 the size of those ones that actually feed on, so there's the larva there, on my midges. And so that's the developing wasp inside of the dead husk of the midge. And so we got 6 different species of parasitoid that just fed on the midge that was on the outside of the other gall. And so hopefully you can start to see these communities are huge, actually, even though they're about that big. So one single gall can host like a lot of species, a lot of different functions, like a species in different niches. So from my view, they're actually pretty important to our ecosystem. They're supporting all of these other species and functions that we can't even tell are happening until we take a closer look. And just one other project, and I'll try not to let it go too long so that there's enough time for questions, but the sort of other half of my thesis was looking into coevolution with the plant. So like I was saying before, this is a back and forth. Like the insect obviously has some sort of influence over the plant to get it to create this structure. But an insect cannot do that to every single plant. It's sort of a lock and key. Like the insect has the key, the right key, the plant is the lock, and they're specific ones that fit together. So many galling insects are specialists. They're completely specialized on either like one genus or a couple different closely related species, sometimes one species, and sometimes one variety of that species. So these flies, all of them were on Ericameria nauseosa, which is rubber rabbit brush. But as any of you might know, as plant people, rubber rabbit brush has lots of different morphologies. Like there's different varieties that you can buy. There's ones that are bright green. There's ones that are the gray kind. And those are different varieties of one species. So in New Mexico, we have a lot of variety graveolens, for example. We have a lot of latticequama. This is taken up at Great Sand Dunes. This is a completely different subspecies, actually, of nauseosa called Ammophila. So there's a bunch of there's diversity within that species of rabbit brush, and it became researchers started to notice that worked on rabbit brush that the galls that they found on the plant seemed to be specific to which variety it was, and so if that was true, that would represent a really, really extreme specialization. So that insect can't even gall all of nauseosa, it can only gall one variety. Um, and so I was interested in looking at this, especially where we have these plants crossing over. Like I was talking about in Rio Grande Gorge State Park, the gray ones and the green ones, these two, are like literally right next to each other and only have one gall on them each. So it's fascinating that they're not crossing. So I took 8 different putative species, so different gall morphologies of this fly that all occur on rubber rabbit brush in New Mexico and Utah and Colorado. Plus I got some from the Pacific Northwest and California as well. So I kind of spanned most of the West with sort of a big gap where I didn't have any friends in the middle of Nevada and in Idaho, unfortunately. So what I wanted to find out is the evolutionary history of these galling flies. Is it explained by the evolutionary history of the different varieties of rubber rabbit brush? And if so, that would be what we call a coevolution. These two things would have evolved together. And that explains why there's such high diversity of gall form and species in this fly group. So I won't go into too much detail because it was a genetic project that took a long time, but I got those 8 different species and I collected plant samples for all of them as well. And made vouchered collections that had the galls on them, so that in the future other researchers could, they'd have two data points in one collection. So I collected vouchers for all of the insects, for all the plants, and then I got genetic samples for all of them as well, and I ended up doing what's called reduced representation genome sequencing, so the whole genome. I took little snippets all across for both the fly and the plant at each paired population. Don't worry about figuring out what's going on here. I'll just summarize for you that we found some evidence that we found evidence that there is some coevolution, and then also some major host switches that appear to be associated with the rabbit brush varieties that are in sandy soils. And so that was sort of interesting, as maybe after the glaciers melted and things began to dry out, these new varieties of rabbit brush probably began to evolve in these drier, more arid soils, and then that resulted in a host switch for some of these flies. But the other cool thing we found is a couple of new species of fly. So from the genetics we found, this one here, which we were calling marshmallow for a long time, I should have put up a new image. But anyway, this is this species here. We were able to describe it as a new species because it not only had a completely different gall morphology, it was on a completely different plant variety that none of the others are on, and the genetics told us it was a different species altogether. And so I got to describe and name that species, which was really exciting. But interestingly, we also threw in this fly because this gall was so different. And we assumed that this would also be a new species, but it wasn't. the morphology of the fly itself had no difference, and the genetics had no difference. So even though these two look completely different in their gall morphology, and their plant is the same, and the fly is the same, they're the same species, but appear to be two, a mutation occurred that created this new haplotype. So we might be seeing a really, really early stage of speciation, which is pretty exciting. And this one is specifically from Eagle Nest. So if you want to go see it, you can go see it up at the state park there. And that's where it occurs. So it's pretty exciting. So I'll wrap it there, but I just, oh, that one is, that's a described species, but it doesn't even create a gall that is external to the stem. In my opinion, it's barely even a gall. It's just a stem borer. And so they are basically impossible to find. And you have to get lucky, basically. But this is it. You can see the little holes. So the larva feeds inside the gall, or the stem in this case. And before it pupates, it chews a little trapdoor for itself, and then pupates. And then when it's an adult, it can just push the door open, because as an adult, it's lost its chewing mouth parts. These adults actually don't eat anything. So it knows that it has to chew its little trapdoor as a larva while it still has its chewing mouth parts. Okay, so hopefully what I've instilled to you is that insect-induced plant galls are unique, they're mysterious, they're diverse, you can identify them, and that they support species-rich and potentially important communities, and are all over the Southwest. I put my contact info in case you ever want to email me about some weird gall, but the best identification resources, there's really only one book. There's so this the California book in other Western states in 2007 was fine, but this is the follow-up from 2021, and this is an excellent book, and it's the only gall book for the Western U.S. So if you're interested, I recommend that book, and anyone's welcome to come look at it. But a friend of mine, Adam, created this website called gallformers.org, which is basically a database of every gall-inducing insect in the continental U.S. and even unknown species. It just has a log as, you know, Rhopalamaia unknown 1, for example. And if you go to that site, you can identify your gall just by putting in the plant species. And so it's especially useful for people who know plants, because you go, oh, I know that's, you know, whatever, Salix bebiana or whatever. You can go in and type in the plant species, and then it'll give you every option of what galls we know occur on that plant species. And so then you can narrow it down from there. So it's a really excellent tool. And then, of course, iNaturalists can also help with identifying galls. But yeah, that's it. Yeah, that's all I have. Okay.